कालच्या व्हिडिओमध्ये पाहिला होता आपण मेल गॅमेटोफाइट, देअर फॉर्मेशन ऑफ मेल गॅमेट्स अँड फॉर्मेशन ऑफ दिस पोलन ट्यूब. आज पाहणार आहोत आपण गायनोशियम, डेव्हलपमेंट ऑफ फिमेल गॅमेटोफाइट आणि female reproductive part. Now this gynoecium, it is the female reproductive part or whorls of the flower. It is also known as the pistil. Pistil, and this is nothing but the similar. Then individual member of the gynoecium is called as carpel or megasporophyle. Individual members, that means they have the single gynoecium or ovary. They have this is known as a carpel. Carpel nothing but stigma, style and ovary. All these part commonly known as the carpel, or it is also known as the megasporophyle. Then जे गायनोशियम आहे ते गायनोशियम दोन टाईपचे आहेत, दिले आहेत. एक म्हणजे एक अपोकार्पस आणि दुसरा आहे सिनकार्पस. अपोकार्पसमध्ये ते कार्पल जे आहेत, कार्पल हे फ्री आहेत. दे हॅव दी फ्री कार्पल्स. सपोज this diagram. Now this diagram will show this. This diagram show the carpels, that's apocarpus, and this one is the apocarpus. These are the separate carpels. Then in case of syncarpus, these ovules are fused. That is the carpels. These are the fused one. This is not the syncarpus. The carpels are united together. Here the carpels are free. There are two type of. Example of apocarpus is mycelia and the syncarpus brinjal. Then this gynoecium consists of the typical part. They have three parts mainly: stigma, style and ovary. This is a single structure. We can say that this one is the carpels. Now this carpel, they having the stigma. It is the outer part where the nectars are present. Then a long tube arises from this stigma into the ovary, and then last portion is the ovary where the ovules are formed or female gametes are formed, and this ovary plays on thalamus. But these are the main three parts: stigma, style and ovary. This is known as the carpels. Now this number of ovules inside the ovaries depend on varies because of in paddy, wheat and mango these are uniovulate. Uniovulate means here is a single ovary and they have the single ovules. This is known as uniovulate. And some plants, the tomato and ladyfinger, they have the multi-ovulate. Multi-ovulate, that means they have the different ovules. This is known as multi-ovulated. Then let's see that the structure of anatropous ovule. Now this anatropous ovule, this ovule is present inside the ovary. Let us say the carpel मध्ये आपण पाहिली ते ovary मधले जे स्ट्रक्चर असते that's the known as the ovules. Now this is the anatropous. Anatropous means the micropyle end or region directly towards the funicle region or lower side, that known as anatropous ovules. Why this happen? We can see that. Now let's see the only structure of this anatropous ovule. This anatropous ovules inside the ovary is attached to the placenta. Now let us see this one is this is the placenta, and these ovules completely attached to the placenta. That's known as the anatropous ovule, or this known as a funicle. Then each ovule develop inside the ovary attached to the placenta. That is a stalk like this known as the funicles, and the place of attachment of this funicle to these ovules is known as a hilum. This one is the hilum. Hilum, it is a part which is a connection between this hilum and the ovules. Then in angiosperm, the most of the common type is a anatropous. This anatropous का म्हणतात त्याला because this end, micropylar end, is directed toward the downward direction. त्याची डायरेक्शन जी आहे ही डाऊनवर्ड डायरेक्शन आहे म्हणून त्याला anatropous ओव्हल. म्हटल्या जाते. Then if we see the structure of these holes, we have to know, we have to see the central part. आपण सेंटर मधून पाहू त्याचं स्ट्रक्चर कसं आहे. This is central part of this ovules. It is known as a embryo sac. This known as a embryo sac. Now this embryo sac, it is surrounded by a tissue that's known as the integuments. Nucellus. This is the nucellus. This embryo sac, it is surrounded by the nucellus, and this nucellus, it is a protective covering. Now outer first covering, this is the nucellus. It is the protective covering. Now then outer to that of this nucellus, the two layers are formed. One is a inner integument, and outer integument. Inner integuments and outer integuments. Now these are two integuments which covers the nucellus, and all this, the function of this is it protect the embryo sac. Centrally it is the embryo sac, then it is surrounded by the nucellus, which is protective covering. Then above this nucellus there is a two layers, that is a outer integuments and inner integuments. There are two integuments, दोन layers आहेत ते प्रोटेक्शन देण्यासाठी. But these two integuments are open at the मायक्रोपाईल रिजन. जी मायक्रोपाईल रिजन असते, ओपनिंग असते, त्या ठिकाणी हा इंटिग्युमेंट्स हा अब्सेंट असतो. ओनली द न्यूसेलस पार्ट इज प्रेझेंट्स and integuments are absent. Then this opening is known as a micropyle. Micropiles, that is a small opening where during the fertilization they are perform the functions. That's micropyle region. Then next they have the first one is a micropyle region, and outer side that's known as a chalazial region. This one is a chalazial region. This micropyle region and chalazial, there are two regions. One is outer side, एक वरच्या साईडचा असतो त्याला chalazial region म्हणतात, खालच्या साईडला आहे तो micropyle region. Two regions. Now this chalazial region presents in a female is the ovule directly opposite to the micropyle. हे दोन लक्षात ठेवायचे. Micropyle region and chalazial region are opposite direction. Both are opposite direction. Then this embryo sac presents inside the ovule centrally. This may be the multicellular structure embedded in the nucellus. That's the then the development. The female gametophytes as female gametophyte development take like that of this male gametophyte. Male gametophyte मध्ये आपण पाहिलं होते की this मायक्रोस्पोर मदर सेल्स MMC म्हणलं आपण त्याला, त्या सेल्स त्याचं डिव्हिजन होते, डिवाइड meiotically to form two tetrads. Now this tetrad gets separated during the dehiscence, and this each pollen grain is formed. That means एका मायक्रोस्पोर मदर सेलपासून 4 पोलन ग्रेन तयार झाले जे आहेत ते मेल गॅमेटोफाईटमध्ये. बट इन केस ऑफ फिमेल गॅमेटोफाईट्स, here is also the MMC cell take place, and this is known as a megaspore mother cells. Megaspore mother cell is undergo meiosis to form the linear tetrad or haploid megaspore. हे जे तयार होते आपल्याला जो cell, that's a megaspore mother cell. First one is the, then this is the first meiosis take place to form the two cells. Then two cell again divide, second meiotic division to form the tetrad. Tetrad means 4 cell formation take place. Now under these three, about these, out of these 4, 3 are the degenerated, and it act as a first cell to the female gametophytes. Then generally one megaspore toward the center is functionally megaspore, and in fact the first cell of the female gamete is undergo successive pre-nuclear division, that's mitotic division. त्यात 8 सेल तयार होतात. आता now let's see the diagrammatic here. Let us see this development of female gametophyte. First cell, it is the female development of female gametophyte. That is the megaspore mother cell. Now this megaspore mother cells undergo first division. The first meiotic division take place and to form the tetrads. Therefore, the four haploids megaspore. These four haploid megaspore cells are formed. Now, after this, what happen? Out of these four haploids megaspore, out of three are degenerate. First three are degenerate and fourth one remain functional. Now काही जणांनी विचारलं होतं की ह्या पहिल्या तीन का डिजेनरेट होतात. बिकॉज ऑफ दिस in female gametophyte there is the only already process occurs, that is apoptosis, program cell death. प्रोग्राम सेल डेथ म्हणजे त्याच्यामध्ये अशी इंटिमेशन दिलेली असते की त्या ह्या सेल ॲटोमॅटिकली डिजेनरेट होतात. त्यामुळे दिस प्रोग्राम ठेवला जसे आपल्या लाईफ स्पॅन राहते सेलचा, तर तो लाईफ स्पॅन हा ठराविक काळासाठी राहतो आणि सेल डिजेनरेट होतात. तशा ते सेल आहे तर त्याचा प्रोग्राम सेल डेथ. आतमधले जे मशिनरी आहेत, आतमध्ये जो न्यूक्लियस काम करत असतो, DNA काम करत असते, त्यामध्ये इन्फॉर्मेशन मिळत असते त्यानुसार ते ॲपोप्टॉसिस होते. ॲपोप्टॉसिस म्हणतो त्याला आपण. ॲपोप्टॉसिस म्हणजे प्रोग्राम सेल डेथ. त्याचे एक पर्टिक्युलर पिरियड झाला की तो सेल डेथ होतात. त्याचं एक रिझन आणखी एक आहे की जेव्हा ह्या फोर सेल असतात, फोर सेलमध्ये सुरुवातीला फर्स्ट सेलचं कॅल्शियम कंटेंट कमी होते, नंतर ते डिजेनरेट व्हायला लागतात. त्यासोबत सेकंड सेलचं कॅल्शियम कंटेंट त्यानंतर लो व्हायला लागते आणि थर्ड सेलचं हे कॅल्शियम ग्रॅन्युल्स कमी कमी होत जातो. आणि जी फोर्थ सेल आहे ते ऑलरेडी फिलअप द कॅल्शियम कंटेंट, कॅल्शियमचे ग्रॅन्युल्स कंटेंट हे फिलअप केलं असते, त्यामुळे ती फंक्शन राहते. फर्स्ट थ्री आर डिजेनरेटेड. नाउ दिस वन इज द मेगास्पोर ऑफ एम्ब्रियो, दॅट्स द हॅप्लॉइड सेल्स फॉर. नंतर ह्या तीन डेंट झाल्यानंतर इथे आपण पाहिलं की मेगास्पोर मदर सेलपासून चार झाल्या, चारपैकी तीन डिजेनरेट झाले आणि एक मेगास्पोर मदर सेल फंक्शनल राहिली आणि त्यापासूनच पुढे एम्ब्रियो तयार झाला. नाउ द नेक्स्ट वन इज द धिस मेगास्पोर एम्ब्रियो अगेन ग्रोथ अँड नरिश बाय द न्यू सेलस. आता आपल्याला न्यू सेलस माहीत आहे. न्यू सेलस म्हणजे एम्ब्रियोशॅकच्या वरची कव्हरिंग जे असते ते न्यू सेलस. त्या न्यू सेलसचं मेन फंक्शन आहे तो इट प्रोव्हाइड द नरिशमेंट और इट प्रोव्हाइड द न्यूट्रियन्स टू दी डेव्हलपिंग मेगास्पोर. नाउ दिस ग्रोथ nourished by these new cells and this megaspore grow and it become larger in size. Then after that what happen, this growth megaspore first mitotic division take place. त्यामध्ये फर्स्ट पहिलं mitotic division, the first mitotic division to form the two cell stage. हे द टू सेल स्टेज, यामध्ये या सेल दोन सेलचं स्टेज फॉर्मेशन झाले. दोन स्टेज फर्स्ट वन इज अ कॅरिओकायनेसिस, सेकंड वन इज अ देन सायटोकायनेसिस. ओनली कॅरिओकायनेसिस, नॉट सायटोकायनेसिस. दिस इज एम्ब्रियो सॅक. ओनली दिस these are the cells develop and two cell stage are formed. First mitotic division, two cell stage. Then second division, it is the mitosis second to form the tetrad. There is four cell stage. जसं आपण पाहतो की पहिल्या मायटोटिक डिव्हिजनमध्ये दोन सेल झाल्या, दोनचे परत डबल होतात, टेट्रॅड फॉर्मेशन होतात आणि टेट्रॅड फॉर्मेशन झाल्यानंतर परत एकदा मायटोटिक डिव्हिजन होते आणि त्याचं एम्ब्रियो शॅक इथं फॉर्मेशन होतं. म्हणजे यामध्ये पहिले आपण तीन मायटोटिक डिव्हिजन होतात आणि 8 सेल स्टेज केले होते. दीज आर द 8 सेल स्टेज फॉर्मेशन होते. नाउ दिस आफ्टर दिस फॉर्मेशन ऑफ द 8 सेल स्टेज, these 8 cells rearrange inside this embryo sac. This is a complete growing of the megaspore, and this megaspore 8 cell stage megaspore. Out of this 8 cell stage, 3 cells goes outer end, that is the chalazal end, and 3 cells goes to the micropyle end. असे वर 3 आणि खाली 3 सेल केले असतात. outer 3 cells is known as antipodal cells. These are the antipodal cell, they have the two polar nuclei, sorry, antipodal cells, and lower sides they having the three one. Out of these three, two are synergids, outer side two are synergids, and centrally is the eggs. Central जे आहेत the eggs आहेत, eggs female gametes. Now these are the three cell stage. Chelazal end having the three cells between the antipodal cells. In the micropyle region have the three cells. Out of three, two are synergids, they are supporting cell, and middle one is a functional gametes. After that, what happens? This stage, after that, the centrally present the two nucleus, that is the, this is known as the two polar nuclei. This two polar nuclei get fused to form this one. Here is a two polar nuclei fused to form one. That means this is a single diploid cells. आता यामध्ये आपण सांगू शकतो की यावरच्या तीन सेल अँटीपोडल सेल, लोअर साइड्स मायक्रोपायल मध्ये तीन सेल्स आहेत, दोन सिनर्जिड्स आणि एक सेल, आणि सेंट्रली एक डिप्लॉइड सेल आहेत. ती 3 + 3 6 are haploid one is diploid cell. That means you can say that here stage is the seven cell, 8 nucleated 7 cells stage. Now here is the complete embryo sac is formed. Here is the complete embryo sac. Mature embryo sac female gametophyte just before fertilization. They have the several nuclei present. 6 are haploid, 1 is diploid. His stage has the 6 haploid cells and 1 diploid cells. That is 7 cell stage and 8 nucleated. This stage is known as the functional embryo sac. This is the embryo sac, and this embryo sac is ready for the fertilization. Now here is we complete the development of female gametophyte.